Our speaker is Dr. Johannes Krause. He's an assistant research professor in the Seagrass Ecology Research Lab at Florida International University. He has an impressive education, especially from many different countries. He has degrees from universities in Germany, in Scotland, and here in the US. He's an expert in seagrass ecology and has some expertise, specific expertise in coastal biogeochemistry and looking at things at the seascape level, which is very important to the project that he will be talking to us about today. So thank you so much, Johannes. I will stop sharing my screen and turn it over to you.
Yeah, thank you very much for the introduction. Let me pull up my slides here. Just a quick check. This is good. All right. Yeah, thanks everyone for joining the webinar. This is going to be a talk on the regional scale drivers of seagrass carbon stocks, and it includes sites at the Nature Coast, but then expands further into Florida and the greater Caribbean. So this is kind of a regional outlook, and throughout this talk, I try to emphasize those points that are specific to the Nature Coast, because that's the focus of this webinar series. So I wanted to start off by reminding ourselves of one of the most critical drivers of environmental change today. We are probably all familiar with the Killing curve, the measurement of CO2 from Mauna Loa in Hawaii. This is a really stark visual and often cited as the first evidence of rapidly increasing CO2 concentrations in the Earth's atmosphere. Now, to put that into historical context, we can add reconstructed data to show the increase since the invention of the steam engine or the beginning of the Industrial Revolution. And going back even further in time, this shows about 2,000 years after the invention of agriculture. So this is to show that the current atmospheric CO2 levels are unprecedented in times of human societal organization as we know it. And if we go back even 800,000 years, way past the origins of our species, we see that CO2 varied considerably between periods of glaciations and warm periods. But even stretching back that far, there have not been CO2 concentrations as high as today. And now the impact of this elevated CO2 on our Earth system becomes more and more clear. And it has been linked to rising temperatures, melting ice caps, sea level rise, and changing patterns of precipitation and droughts, more intense droughts and heat waves, flooding, wildfires. And in Florida, important, of course, tropical cyclones that may become more intense due to higher water temperatures. So it is clear that the conditions on our planet are rapidly changing, and this will have effects on the species living on it, including us, that we yet have to fully understand. Now, carbon is always cycling through the atmosphere, the hydrosphere and the geosphere, and human activity is affecting the balance of carbon cycles, mainly through the extraction of fossil fuels. To understand how the carbon cycle and our climate are changing, our climate scientists are measuring and modeling all aspects of it. And while it is relatively easy to measure human-made CO2 emissions, the flow of carbon through natural systems like land and ocean remains really one of the least understood parts in climate science. So there's a huge number of processes that can be very complex in the natural world. And that means that there are still important gaps in our understanding. So as researchers and perhaps practitioners interested in coastal vegetated ecosystems, we are focusing on this part of the system that I highlighted here. Now, the general idea is that vegetated coastal ecosystems like mangrove salt marshes and seagrasses are taking up more carbon via photosynthesis than they lose from respiration. And a part of that generated organic matter is buried in the soils. Now, in coastal vegetated ecosystems, those soils can be waterlogged. And that leads to little of that organic matter being respired and returned to the atmosphere. So the resulting soil carbon stocks can be really high and especially large compared to terrestrial systems. The highest stocks that we know of are in mangrove soils, but seagrasses were also found to store a lot of carbon in the soils. Um, this graph on the left that I show over here is from Fogren and others in 2012, which was really the first global synthesis of seagrass carbon stocks. And what they show here, I think, is really this large variability in seagrass soil carbon stocks. So they can be as high or higher than in mangroves, but they might also be very small. This is just a massive variability that is or was not that well constrained, at least in 2012 when that synthesis happened. Now, when we are talking about blue carbon, it can also be very helpful to think about the timescales that are at work. Because when extracting fossil fuels, we are moving carbon that was buried for many thousand years or even millions of years and is fossil carbon. And we move it to the modern carbon cycle. And now, blue carbon is about the portion in the modern carbon cycle that sequesters carbon from the atmosphere and into coastal vegetated ecosystems, the vegetation itself and the sediments. And leaves and roots, this vegetation, typically has a relatively short turnover time. So the carbon stored in those compartments is there maybe for like a few years or so. While the carbon that is stored in the soils of those ecosystems can be there for hundreds or maybe thousands of years. But so blue carbon is really about short-term remedies, geologically speaking. So we want to be sure that coastal vegetated ecosystems do not get lost and their blue carbon return to the atmosphere. Ideally, we want to restore blue carbon ecosystems and move a portion of modern carbon from the atmosphere into the coastal ecosystems. But that is to say, as long as we keep adding ancient fossil carbon into the modern carbon cycle, we're kind of fighting an uphill battle against climate change. So my talk today is really focusing on the modern carbon cycle and the apportioning of carbon in those different compartments. On this map by Dan Fries and others, the green circles are current planned mangrove projects that aim specifically at blue carbon. This either by the protection or the restoration of blue carbon habitats. And there are also some blue circles on this map, and those are seagrass specific projects. So at least when this study was published, there were really few seagrass projects compared to mangrove. And this is partially due because for mangroves, we really have a good idea of carbon stocks and burial rates, and they are also relatively easy to measure because they are mostly above the water. For seagrasses, this variability is so large and measurement is relatively complicated. And so not that many projects were proposed as of today. In order to maximize the impact of conservation action for blue carbon, we need to know what types of systems sequester and store large quantities of it. And this is especially important for seagrasses because they can vary so much. There has been a lot of recent research, though, focused on uncovering those controls and to minimize this uncertainty. What I show you over here is a graph from Ines Mazarasa and others that show the drivers of blue carbon in seagrasses. And for example, they show that species composition is important, canopy complexity can play a role. The landscape, how the seagrasses are arranged, is potentially also important, as are trophic interactions. These are just some of the points that they made. There are many more. And then just a couple of years ago, Hilary Kennedy and others published a study using a global data set of seagrass carbon stocks. And they tested whether seagrass traits would be important in determining how much carbon is stored. And sure enough, they found that some seagrass traits are important. For example, how many leaves there are per shoot. They found that below and above ground biomass plays a role. Then also the lignin content of leaves and so on. They also found other environmental controls. For example, they found that in lagoons, you would have higher carbon stocks than on reefs. But they really have a good evidence for seagrass traits playing an important role here. And then most recently, I've been working with the Blue Carbon Initiative to further constrain global seagrass blue carbon estimates and look at the drivers of carbon stocks. And we found that the different types of seagrasses are also associated with different carbon stocks. So we categorize seagrass species into functional groups depending on the life cycle. Persistent seagrasses, in the Caribbean, that would be Thalassia, for example, that form these large rhizomes and are kind of the climax species of the ecosystem. We group them together, and then more ephemeral ones and intermediate types of seagrasses. And we contrasted those and found really that those persistent seagrass meadows, they do have larger organic carbon stocks than seagrass meadows of ephemeral or intermediate species. In fact, those ephemeral species, that would be, for example, Halophilas or Rupia, their carbon stocks didn't seem to be larger than unvegetated flats. So potentially some of the seagrasses might not even increase carbon stocks at all. We also looked at different marine ecoregions. And this is, of course, a little bit influenced by data availability. In this map, all of those dots, those are the sites where we have soil cores in seagrass meadows. So that's all the data that went into this work. And we still have a lot of gaps. The Caribbean is okay covered. But on the other side, the Eastern Pacific, for example, is a large gap. It would be great to have more data from Western Africa, perhaps India. But I show you this because the tropical Atlantic really came out as a hotspot for seagrass carbon stocks, globally speaking, at among the highest average and median stocks. So now to better understand where there are large seagrass organic carbon stocks in the Greater Caribbean, including Florida and the Nature Coast, the Nature Conservancy funded the Caribbean Carbon Accounting and Seagrass Project, or short CariCAS. And the goals of this project were to assess the variability and the environmental drivers of seagrass organic carbon stocks, and then second, to map carbon stocks across the region. And that project basically wrapped up last summer. And today I will talk about our work addressing the first objective. So the overall idea was to devise a methodology to assess seagrass carbon stocks and the environmental covariates of it, and then build a regional network of seagrass researchers who would be interested in implementing this method and then building up a large database and analyzing it together. So on the right side over here, you can see a sketch of our survey design. The idea was that we would take a sediment core up to 1 meter depth, and then that would be the center of our survey. And around it, in a radial design, we would have 16 quadrats where we would measure all kinds of seagrass meadow aspects. For example, the abundance of each of the species that are there, abundance of other benthic organisms such as sponges and algae, coral, and so on. We will get an idea of the seagrass species composition for that. And at half of those, so 8 of the quadrats, we would also take biomass cores that allow us to calculate above and below ground biomass as well as shoot densities. And all of that is species specific. Then from the sediment core, we analyze different core intervals for organic, inorganic carbon and nutrient contents, so nitrogen and phosphorus, as well as stable isotopes of carbon. And we would also collect seagrass leaves to then in the lab analyze for carbon, nitrogen, phosphorus contents and stable isotopes. So all of this is building a really rich data set. And these different things that we measure, we think we can relate to environmental drivers that are affecting them. And by analyzing those, we hope to get a good idea of which environmental drivers are controlling carbon stocks in the region. Now, we reached out to as many organizations throughout the region as possible. That included NGOs, universities, government institutions, and all in all, we built a network of 27 partner organizations who took part in the project and sampled at least 2 seagrass meadows in their area. Many of them sampled more, and this resulted in data from 21 countries, 27 partner organizations, collecting 76 sediment cores, almost 600 biomass cores, over 1,500 core intervals, subcores, and almost 1,200 georeferenced quadrat locations that record all of these metrics that I showed before. Now, in this map, you can see the spatial distribution of our sampling sites, pretty much covering the Caribbean Sea, the basin, outlining it, and then stretching into the Gulf of Mexico. So the aim of this project was to sample the greater Caribbean, hence sites in Florida included. The Nature Coast sites are not actually the northernmost here because we also sampled Bermuda. As many of you know, Bermuda, the marine ecosystems are very comparable to the Caribbean. They have the same seagrasses there, similar fishes, and so on. So it's really part of the same system. But Nature Coast sites are among the northernmost and perhaps furthest from coral reefs in this data set. Now, I wanted to shout out all of the partner organizations and collaborators that we had for this project. And yes, since I'm here, thanks especially to Dr. Reynolds and Dr. Barry of the UF-IFAS Nature Coast Biological Station. I saw that some partners from other organizations have joined this webinar as well. So thank you to you as well. So I'm really incredibly thankful for everyone who made this project happen. And it's been really, really cool to experience to be part of such a large project where so many people care about seagrasses in the same region. That's really kind of inspiring. Yeah, let's jump into the findings. First, I wanted to start with seagrass biomass. So we can also consider seagrass biomass as a carbon stock because there's organic carbon stored in the sediments and then in the living biomass. But of course, biomass is much smaller than what's stored in the sediments, as I showed you earlier. But we did find that there are clear differences in the amount of above and below ground biomass depending on the seagrass species. And note that the y-axis here is on a log scale. So generally, all of the seagrasses have larger biomass in rhizomes and roots compared to leaves. So if you're in a seagrass meadow, really the above ground part, those leaves, what you see, that's the smallest carbon store there is. If you go into the sediments, look at roots and rhizomes, that's where most of it is. And maybe it's also interesting to see that the invasive Lophola stipulaceae, which we had at quite some of the sites that we surveyed, had the second highest average above ground biomass of the species we encountered. So there can be really a lot of carbon, relatively speaking, stored in this species. Now, on the Nature Coast, maybe you don't care about Halophila stipulaceae all that much yet, but it's been only a few months ago that for the first time we found it in southern Florida, in Miami. So at one of the marinas over here, it is growing and the invasive seagrass is spreading, it's establishing itself. So it's probably only a matter of time for it to spread around Florida as well. Just a heads up. But as I showed you before, the seagrass meadows really store most of carbon in their soils. And these over here are the carbon stocks of all of the sediment cores that we collected. So to compare between sites, we typically only count carbon to a specific reference depth. And it differs, different studies use different reference depths. Here the sites are sorted by their carbon stock in the top 30 centimeters of soil. And that was done because that is a depth that all of the cores reached. And Not all of them necessarily reached 1 meter depth, because seagrasses don't always grow on a meter of soil. There can be less of it. And perhaps sometimes you lose a part of the core and then you don't have the entire meter. But in this graph, the solid portion of those bars, that's the 50 centimeter reference depth, and the bright shaded part, that would be the 1 meter. We did not extrapolate, so that's a practice that some researchers do, or practitioners, where you take a smaller portion of a core, say like 30 centimeter core, and then assume a linear carbon concentration with depth and extrapolate to a meter. We didn't do that here because what we found is there's so much variability going on that just assuming and extrapolating is not really precise. And so what I want to show over here is that generally there's a very large variability. The median 30 centimeter carbon stock that we had in this data set was really much higher than the global average or the global median. So this really confirms that the Caribbean is really a hotspot for seagrass carbon stocks. All of these sites, they are sorted left to right by the magnitude of the 30 centimeter carbon stock. And I just point out the two nature coast cores that we have in this data set. So on the left, that's a site by Crystal River, and on the right by Weeki Wachee. And even though they are like relatively small, relatively close together, they're quite different, with Weeki Wachee being one of the lowest carbon stocks in the data set, and the Crystal River site, one of the largest. But that's for the 30 centimeter stock, right? But like, if you are considering now the entire stock, the 1 meter stock, the Crystal River site is actually among the sites with the highest stocks in the entire data set. So you can already appreciate, I think, that there's a difference depending on what reference depth we choose here. If we're talking about 30 centimeter stocks, Crystal River is kind of in the upper third. But if we talk about the 1 meter stock, it would be perhaps the third highest there is. So that's due to this organic carbon not being distributed evenly down core, but there are big differences. Now, these are the core profiles from Crystal River and Weeki Wachee, and we show organic and inorganic carbon, as well as nutrient concentrations and dry bulk density. So typically, organic carbon concentrations are highest at the surface, because that's where the organic matter is deposited, and then they decrease with depth as the labile part of organic matter is decomposed over time. And at some point, you would have a constant concentration of organic carbon. That's what some people call the burial concentration. And that's when you don't have any labile organic carbon anymore that can be remineralized by microbial activity and so on. So that's really the portion that stays in the sediments for a long time, potentially. Then dry bulk density usually does the opposite of organic carbon. It increases with depth up to a certain point as the really as the soil consolidates over time, so it becomes denser down core. Nutrients like nitrogen, phosphorus, they're typically covary with organic carbon because all of that is bound in organic matter. Organic matter is really determining nutrients as well as organic carbon. But what you can see in Crystal River is that there's a deviation of that typical profile. So there we have higher organic carbon concentrations below 60 centimeter depth. And this is the reason why the 1 meter stock is so high there, even though the 30 centimeter stock is not as high. So this deep portion of the core with a high carbon, that could indicate that there were past changes in ecological processes. For example, it could be a past transition from mangrove or marsh to seagrass habitat. We are now analyzing those samples for carbon stable isotopes, and those can tell us about the origin of organic matter. So that's going to be very helpful in determining whether there was a switch in habitat type. These are two examples from other CariCAS sites. On the left is Bahia Honda from Colombia. On the right is Lagoon Park from Bermuda. And here we already have carbon stable isotope data. So seagrasses and mangroves, they have different ways of fixing carbon into biomass. And there's an isotopic fractionation associated with that. That results in mangroves typically producing organic matter with a stable carbon isotope ratio of about -28 per mil, while seagrasses typically have a carbon stable isotope ratio of -10 per mil. And in these core profiles, the -10 and the -28, they're indicated by dashed lines. The purple is the carbon isotope ratio of organic matter, and the green is the carbon content again. And so what you can see over here in these cores is that you have these deeper layers with very high carbon contents that have a stable isotopic ratio consistent with mangrove material. And then towards the top of the cores, really much lower organic carbon contents, and the signature of that is much closer to that of seagrass. So those are most likely transitions of habitat where at some point in the past at that location we had mangroves, and they laid down this mangrove peat, really high organic carbon. And then over time, probably as sea level rise, mangroves get replaced over time by seagrass. And now the seagrasses, they sit on this mangrove peat, resulting in really large carbon stocks. We can look at that in another way as well. So here are all core intervals in our data set for which we at the moment have carbon isotopic ratio data. And you can see there is this group on the left upper part of the graph where really the high carbon content data, those are associated with very low carbon stable isotope ratios, again, showing the signature of mangrove peat. So we use these cores with mangrove peat to do some radiocarbon dating. And we found that those mangrove layers, they were 2,000 to 6,000 years old. Again, likely that's about the time when the area was flooded more. Sea level rise led to a transition from mangroves to seagrass habitat. But since then, everything that accreted on top of it, so all of the soil that is sitting on top of the mangrove layer up to the sediment surface, that means that all of that has been deposited since 2,000 or 6,000 years ago. And in the table, I show what kind of sediment accretion rates and carbon burial rates are associated with that. And as you can imagine, over that long time scale, those rates are really, really low. So in these cases, we have really low accretion rates leading to very low carbon burial rates. But the carbon stocks, they are still very high because of this buried mangrove peat. So on the flip side of that, there may be seagrass meadows in our study that accrete a lot of sediment, but if the organic carbon concentrations in the sediment are low, the stocks are going to be low. But that doesn't mean that they bury little carbon, because due to high accretion rates, the carbon burial rates may actually still be very high. We would not know that because we didn't measure specific burial rates in all of the cores. But it's important to keep that in mind because, yeah, carbon stocks and carbon burial rates, they're really two different things. So in the context of seagrass conservation, we would want to target different types of seagrass meadows depending on whether we care about avoiding emissions by protecting existing carbon stocks, or whether we want to enhance the future burial of organic carbon, aiming at meadows that have high burial rates. Now I'm going to switch gears a little bit and look at other environmental drivers of carbon stocks in the greater Caribbean. So we tested our data set for other covariates of carbon stocks. Here we checked whether there was a difference in between those different seagrass species that we encountered. And generally, there was a tendency towards higher stocks in Thalassia-dominated meadows, but there were also a lot of Thalassia meadows that had very low stocks. So in the end, there was not really a significant difference between meadows dominated by Syringodium, Halophila, or Thalassia. What's interesting maybe is that the meadows that we had in our study that were dominated by halodole, they seem to have much higher organic carbon stocks than the other meadows. But it's a pretty small sample size. I think we only had 3 or 4 halodole dominated meadows. We also tested whether seagrass traits would affect carbon stocks, and we did not find a relation between above ground biomass and carbon stocks, and neither between below ground biomass and carbon stocks. So this is something that Kennedy and others found in global data, as I showed you in the beginning, but across our sites in the Caribbean, that doesn't play out. We did, however, find a positive effect of canopy height on organic carbon stocks. So there is probably an effect of overall productivity in the system. It's also possible that higher seagrass canopy may help increase deposition by trapping particles in the water column, leading them to deposit in the seagrass meadow. We also found that across the Caribbean, there were larger carbon stocks associated with soils of finer sediment size. This is something that's generally reported in marine sediments, seagrasses in particular. Small grain-sized soils, they have a higher relative surface area that might help protect and bind organic carbon and make it harder to remineralize this. So maybe it's protected from microbial activity a little bit. It's also true that grain size tells us something about the energy environment of a site. So if we have a lot of fine material, muds and so on, they usually settle out in calm water. While the coarse sediments, you usually have them when there's a lot of hydrodynamic energy. So this finding could also mean that generally sheltered environments are more conducive to higher carbon stocks than high energy environments are. And lastly, we also found that connectivity of different blue carbon habitats may play a role in carbon storage in the Greater Caribbean. So we found that those seagrass meadows that are closer to mangrove habitats and those that had more mangrove habitats, a larger area of mangrove habitat close by, that those would be associated with higher organic carbon stocks. And this is different from what I showed you before with the buried peat, because the buried peat, that's like deeper in the sediments. But what I show you over here are the top 30 centimeters. Those surface carbon stocks, they are also higher when mangrove is nearby and when more mangrove is close by. So this probably has to do with facilitation between different blue carbon habitats. There's really the seascape idea that we don't look at one habitat type in isolation. And it's likely that lateral fluxes of organic matter from maybe exported from mangroves, they might supplement the seagrass carbon stocks. So again, through stable isotope analysis, we may be able to connect those ideas and see whether those surface sediments with higher organic carbon stocks are more mangrove-like. But we're still waiting on some of the stable isotope data. Now, to bring it all together, we did some modeling. We used random forests, multiple linear regression, and partially squares regression to predict organic carbon stocks from all of those attributes that we measured. And really all of these different modeling techniques, they all came out sort of on the same important variables. So all of them pointed towards the importance of mangrove habitats, seagrass canopy height, and then the fine sediments. Overall, those explained a pretty good portion of the variability in the data. On the right, I show the most important variables that came out from partially squares regression, and I try to color them by variable type, so that the teal would be seascape metrics, blue has to do more with the benthic cover, and maybe pink is morphology and biomass. And just to say that really seascape metrics seem to be pretty important in this data set. So across the greater Caribbean, including the nature coast, Florida, so on, seagrass carbon stocks much better predicted if we take into account the seascape configuration and other blue carbon habitats nearby. So we can combine our results with studies that have been published before and describe a conceptual model of the drivers of seagrass organic carbon stocks. So generally speaking, the organic carbon is generated through primary productivity in the seagrass meadow and can as well be imported via lateral fluxes from adjacent ecosystems. And then it is buried. And this burial depends on sediment deposition as well as seagrass traits. And then some of that buried organic carbon is remineralized and lost again to the atmosphere. And that seems to really depend on the energy environment, causing erosion, resuspension, as well as the sediment type. So how much fine sediment do we have? And of course, microbial activity, which is breaking down the organic matter. And in addition to these current processes, the past is also important. So the ecological and disturbance history of a seagrass meadow can also affect the carbon stock, as we've seen with those examples of the buried mangrove peat. But by taking into account all of these compartments, we can have a pretty good idea of carbon stocks and seagrasses in this region. All right, so I think I will end it here, and thank you for your attention. Many thanks to all the collaborators and funders, and also to the Nature Coast current series for having me. And if you're interested in the field and lab methods this project developed, you can check out our website. The illustrated guide is published as a DOI with a 2. Yeah, and feel free to always send me an email with any questions.
Thank you so much. That was a lot of information, a lot of cool information, and I learned a lot. So if anyone has questions, feel free to type them into the chat, or if you would like to unmute yourself and ask that question, you're welcome to do so. I think people might be a little busy filling out their poll, so I can ask that first question. I really liked the way you really clearly illustrated how important conserving these carbon stocks and conserving these really productive seagrass meadows is, as opposed to restoring seagrass meadows to get carbon. I appreciated that, and I appreciated the data that you had behind that. I was wondering if you could comment, and this may just be that we don't know the answer to that, but when we're thinking about these really important seagrass meadows that are storing lots of carbon that might be degrading, how long do we have to turn that around? Like, is it decades? Is it a couple of years? Like how vulnerable are we?
Yeah, that's an excellent question. So to turn it around in terms of stopping the degradation of a seagrass meadow, or to recover the stored carbon, because those are kind of different things and are both pretty important. Maybe the latter part of protecting the seagrass meadow and then restoring the amount of carbon that is in the sediments versus a seagrass meadow that was never degraded or damaged. That's a really tricky question, and it seems that there can be decades passing before organic carbon stocks are recovered in sediments, which also has to be kept in mind when you want to restore seagrass for the purpose of carbon. In terms of stopping the degradation of seagrass meadows, yeah, there have been these large-scale studies looking at seagrass meadows worldwide, and there was a very famous one in 2009, I think, by Michelle Weikert and others, that really set the tone by looking at long-term change and finding that there's something like a 2% per year loss or something like that. More recently, Gillian Dunick did another study, and she found that in some areas of the world, specifically those that invested a bunch in ecosystem protection, those trends have been reversed or at least stopped. With the example of Europe, for example, it looks like there's no net loss occurring anymore at the moment. But yeah, globally, there's a lot to do. And there's, of course, different interests in using the ocean. And as people who are interested in keeping the seagrass there are just a portion of them, right? Oftentimes there's conflict with other uses such as fisheries, which seem to be yeah, a major problem. But on the other hand, yeah, people got to eat fish.
Yeah. Well, we have a hand raised. Fenella, do you want to ask your question?
Hi, yes. Great work, Johannes. It was super relevant and I learned a lot. I was wondering about, I think you had a graph in there about carbon stocks based on like Thalassia dominated seagrass beds, and like the results were very widespread. Like it wasn't just they were the most highest, but they were also like in the lower phase as well. Yeah. I think, yeah, that one. I just was
Mhm.
wondering if you had any other hypotheses on what factors could be impacting the carbon storage in the Thalassia dominated beds that would make them be so wide.
Right. Yeah. Yeah. I think in the Greater Caribbean, Thalassia is really growing in so many different types of habitats, right? If you think about the other factors that I showed that influence it, for example, the type of sediment you have and the canopy height, so productivity of the system, Thalassia can grow in all kinds of those circumstances. So it is really good at growing in low nutrient environments, those oligotrophic areas where you have very little nitrogen, phosphorus, both of them. And that's one reason why it can outcompete the other seagrasses. But so you would have, for example, back reef areas with those Thalassia meadows that are in this like coarse shell type sediments, and you really have low carbon stocks there. I think those are typically the ones that you see at the lower end of this spectrum. So just overall, the seagrass is able to be in circumstances where other seagrasses are not that competitive, but those circumstances are not really helpful for accumulating a lot of organic matter because those systems just don't have much. It's more reef dominated, a lot of carbonates and so on. But on the other hand, you can also have Thalassia growing in those super productive lagoons, maybe next to mangroves. And that's then the higher part of those of that range of Thalassia I show over here. So to make that short, I think it's the versatility of the plant and the different environments you find it in. So this is to show that it's not really the species that affects the carbon stock so much. It's rather the circumstances it grows in.
Awesome, thank you.
We have a question in the chat that you kind of just addressed, maybe accidentally, but I'll ask it anyway in case there's something else you want to add. You showed that reef area was an important factor. Can you comment whether that's a positive or negative relationship? Does having more reef area near your seagrass increase or decrease your sediment carbon content?
Yes, thank you. Yeah, I guess I alluded to it a little bit. Yeah, so it looks like there's a relationship in a way that, yeah, the highest carbon stocks are further away from reef. Not sure it's the reef itself that is doing this, rather the fact that those lagoons with high carbon stocks by themselves are typically further away from the reef. And if we take cores on the reef, those sites are yeah, they don't have such fine sediments, more large carbonate parts and so on. But it's not that we found a linear relationship. And it's partially, I think, due to the nature coast sites because they're really quite far from the reef. But as I've shown at Crystal River, you have pretty large carbon stocks still. Um, Whereas at Wikiwatcher, you have low ones, even though it's far from the reef. So it's not that straightforward. There's a whole other dimension to blue carbon, which is the inorganic carbon cycle. I didn't mention that in the talk, but I will just say here, if there is carbonate production in a seagrass meadow, for every mole of carbonate that is produced in the water, some CO2 is released to the atmosphere. And so what some people do, what we should do is also taking account of carbon production, and that offsets blue carbon in some circumstances. And so, especially at a reef where there might be a lot of carbon production, that is an additional factor that might lead to more CO2 being released to the atmosphere versus buried in the system, this whole carbonate cycle. I didn't address that here yet, but it's a dimension to keep in mind too.
Great, thanks. And a couple more questions in the chat. The next one is, so is it your opinion that these findings strengthen the importance of maintaining connectivity between seagrass meadows and adjacent habitats?
Yeah, absolutely. Yeah, I think we are having this kind of a little bit of an isolated approach, perhaps that's part of how academia worked or works to a degree. We have all these experts that are seagrass experts or mangrove experts, and we think of those boxes a little bit separately or trying to figure things out in one habitat. But nature doesn't think that way, and I think a lot of data actually shows not just this. That those systems are connected and they are facilitating. And so, yeah, my opinion is that it would be great to think about them holistically. So now I'm not a spatial planner or anything like it. So I'm sure there are problems associated with it. For example, I know that historically mangroves have been managed by terrestrial folks, forestry folks, versus seagrasses would be in the marine realm. So it might be complicated to think about them in the same context. But ecologically speaking, I think, yes, they should be considered in tandem.
I'm going to skip one question because there's one that's sort of related, but I'm going to come back to your questions if you notice that I skipped it. The next question is, super cool work and great presentation. Really interesting that you found some potential facilitation between mangroves and seagrasses. Are there next steps further exploring these dynamics?
Yeah, for this project, there are currently no next steps in that direction. We are still waiting on a lot of carbon stable isotope data, and that will likely result in a study that is using this data set to specifically look at the mangrove seagrass aspect, but no more data collection in that regard. Now the project is pivoting a little bit more to the mapping side of things, and that's not super related. But yeah, I hope that there's going to be more research by folks in our network and outside of our network, really pointing out these connections.
Yeah,
Thanks.
these next two questions, you teed those up really well too. And the next one is, I realize that this will depend on funding, but are there future projects that you think the CariCAS network should work on next?
Yeah, excellent question. So funding cycles are always heartbreaking because at some point you got to stop and wrap it up. And specifically with a project like this, where you have great interaction of all kinds of different partners, it would be a shame to not be able to follow up. So we are actively looking for funding sources to make follow-ups happen. Because in between the different researchers of this project, there have been so many great research questions that were developed that are applicable to the entire region, and that would be great to address them using a network like the CariCAS network. For example, the question of sea water temperature rise across the region, how's that affecting seagrasses and potentially carbon? Of course, the spread of the invasive Phytophthora stipulaceae. I mentioned that a little bit, but this is an ongoing question. And specifically in relation to ecosystem services. There's a lot we don't know about the seagrass when it's replacing natives, what is happening to fish communities, what is happening to carbon storage, and so on. So a lot of questions that would be great to address with this network. And yeah, if you're in a funding agency and look for projects to support, write me an email.
Yeah. Well, there's another question that maybe is an email too, and I can follow up and connect you guys if you don't know each other. But there was a Lauren Hall noticed that there were no sites on the east coast of Florida, and wanted to know if you're looking for collaborators there. And then
Yeah. So,
and then I think
yeah, so
go ahead.
go for it.
I was going to read the last question because we're almost out of time. And that was to build on the reef distance carbonate topic. Could seagrass productivity closer to the reef be higher if CO2 is more readily available for photosynthesis due to the reef's carbonate precipitation, leading to greater carbon burial?
Yeah, that's a great thought. So I'm not an expert in CO2 availability and how far seagrasses would be limited in their productivity and growth by availability of bicarbonate, I guess, and CO2 in that regard. But yeah, there are some great folks working on this. It plays into this idea of importing, exporting alkalinity as well, and what that is doing to productivity, calcification, and so on. But yeah, I should probably not comment on that because I just don't know enough about it. But it's a cool idea.
Yeah. Awesome. Well, I think we have used up all of our time with some great questions. If anyone has any questions that we didn't get to, I'm sure Johannes would be happy to answer those over email. Thank you so much for being here. That was a really great talk. Thanks for everyone for showing up so close to the holiday. We will continue Nature Coast Currents in 2025, and our next one will be January 15th, where we'll hear about some oyster reef restoration. So thanks so much and have a good break, y'all.